Hello students, welcome to biology class. So today we're going to discuss about translation process. So far we were discussing about central dogma. What is central dogma? DNA is transcribed into mRNA. mRNA is translated into protein. So we're going to discuss more about translation process. What is translation? The biosynthesis of a protein or a polypeptide inside a living cell. In process of translation, the language of nucleotide sequence of mRNA is translated to the language of amino acid sequence. Where it occurs in cytoplasm where the ribosomes are located. You know one thing, it is a universal process. The translation process will be the universal process. So we're going to discuss more about this. For the creation of new generation, the genetic information stored in DNA has to be transferred from one generation to another. The process begins with the replication of DNA followed by transcription and translation. Let us have a discussion on translation, that is the protein synthesis and the structure of tRNA. In molecular biology, the term translation can be defined as the process of converting nucleic acid information into amino acid. So what is nucleic acid? DNA and RNA. It also relates to the process of producing protein from mRNA templates. During the process of translation, sequence of nucleotide present on the RNA is translated into amino acid sequence of proteins. The complete reaction of translation is carried out by ribosome, where both ribosomes and tRNA dock on a matured mRNA transcription and select multiple enzymes in an energy to intensive process that uses ATP, adenosine triphosphate. The structure of tRNA. The DNA is transcribed into mRNA on the basis of the complementary. Translation is a process of conversion of nucleic acid information into amino acid. There is no complementary between amino acid and mRNA. Hence, translation is not controlled by complementary but the genetic code. Since amino acids cannot read this genetic code, they are dependent on an adapter molecule. So this adapter molecule is called tRNA, that is transfer RNA. So here I want to ask you a question. So what are the types of RNA? tRNA, mRNA and sRNA. Structurally, the tRNA is an inverted L-shaped molecule which has an anticodon loop and amino acid acceptor end. The anticodon loop makes bases complementary to the codes on the mRNA. And amino acid hence bind to the respective amino acids. Thus, it helps in protein synthesis. Each amino acid has a specific tRNA, initiated tRNA, which initiates the translation, while stop codons have no transfer tRNA. So in this slide, you're going to study about the structure of tRNA. So this is the structure of tRNA where it has acceptor arm, variable loop, anticodon loop, D loop. So what was this part indicates? We're going to study in detail. tRNA or transfer RNA is a type of RNA which helps in the synthesis of protein from mRNA. tRNA function as an adapter molecule during the translation process. So where here it is adapter molecule, yes, this adapter molecule is known as soluble RNA, that is sRNA. As an adapter, it links the amino acid to nucleic acid. It carries the amino acid to be added in the peptide chain and also dissipate the codon for the same in the mRNA molecule. So let's learn about its structure. tRNA structure. The tRNAs are generally 76 to 90 nucleotides long. So when you count here, there will be like 76 to 90 nucleotides. Each amino acids are specific tRNA. Stop codons are not recognized by any tRNA. The secondary structure of tRNA, it looks like, we just look, no, how it looks like. So the shape of the tRNA, it looks like clover leaf, and tertiary structure is like an inverted L shape. The folded structure is formed due to hydrogen bonding between complementary bases. Now we're going to discuss about acceptor arm. It is formed by the base pairing of 7 to 9 nucleotide of 5' terminal and 3' terminal. The 5' terminal has a phosphate group and 3' ends with a specific sequence of CCA or CCA tail. The amino acid attached to the 3' which bears hydroxyl group of the acceptor arm. So the aminoacylization of tRNA or charging of tRNA is the first step of the translation process where it reacted in acceptor arm. So these enzymes, aminoacyl tRNA, it's synthesized to catalyze the reaction, to induce the reactions, okay? So next we're going to discuss about the D loop. So D loop here on the left hand side of the picture. D arm has a stem of 3 to 4 base pair and it ends in a loop called D loop, as it is generally contains dihydrouridine, a modified nucleotide. Next we're going to discuss about anticodon loop. So this will be the bottom structure of tRNA. It has a 5 base pair which bears like a long stem. It has an anticodon loop which contains a complementary codon which contains 3 nucleotide sequences. Present on mRNA for the amino acid it carries. So these unpaired base of anticodon loop pair with the mRNA codon. Each codon is identified by a specific tRNA loop. Next we're going to discuss about T-arm consists of stem of 4 to 5 base pair and a loop containing pseudo-uridine modified uridine. Next we're going to discuss in detail about variable loop. So variable loop is present between the T loop and anticodon loop. Its size varies from 3 to 21 bases. It helps in the recognition of tRNA molecules. Next we're going to discuss about the tRNA function. It plays an important role in protein synthesis. It acts as an adapter molecule for linking amino acid to a specific codon present in mRNA. Aminoacylization of tRNA is the first step in protein synthesis where it will be like the initiator 1 to synthesize the process. tRNAs are specific to each amino acid and carries them during the translation process in the ribosomal subunits. So in further slides we're going to discuss about the ribosomes and what will be the sizes. Yes. The tRNA transfer the amino acid to the growing polypeptide chain in the ribosomes, which has 3 binding sites for tRNA, namely A, P and E, which correspond to aminoacyl, peptidyl and XII. So what was the key here? A, P and E, where it denotes aminoacyl, peptidyl and XII. So this decoding of codons of mRNA by specific tRNA continues until the entire sequence for a polypeptide chain is translated. Components of translation. So the main components of translation include mRNA of both eukaryotes and prokaryote. tRNA, it helps in sending commands or transferring the information between protein and nucleic acid. Ribosomes, it contains ribosomal RNA and protein. Therefore, it is called the manufacturing unit of cell. Enzymes, here it is required for the formation of peptide bond between the amino acids and also to attach amino acids to the accurate tRNA molecules. Next, protein. So they are the soluble factors which are required for proper initiation, elongation and termination of translation. So this will be the step are included in the process of translation. So now we're going to discuss about the protein synthesis. This is a process which is carried out by translation. The process by which the mRNA codes for the particular protein is known as translation. In the process, the ribosome translates the mRNA produced from DNA into a chain of specific amino acid. So this chain of amino acid leads to protein synthesis. It is a process where the expense of ATP is required and this energy is given by the charged tRNA. So already we were discussed about tRNA structure and functions. So the whole machinery of translation is present in the ribosomes. The biosynthesis of protein or a polypeptide inside a living cell. In process, a translation, the language of nucleotide sequence on mRNA is translated into the language of amino acid sequence. So what was the process happening here? The translation of languages like the nucleotide sequence on mRNA is translated into the language of amino acid sequence. It occurs in cytoplasm where ribosomes are located. It is a universal process. In translation, messenger RNA is decoded to produce a specific polypeptide. This uses mRNA sequence as a template to guide the synthesis of chain of amino acid that form protein. So many types of transcribed RNA such as tRNA, rRNA and sRNA are not necessarily translated to amino acid sequence. The ribosome consists of a bigger subunit and a smaller subunit. The larger subunit in turn consists of two molecules of tRNA that are placed close so that peptide bond could be developed at the expense of sufficient energy. The mRNA enters a smaller subunit which is held by the molecules of tRNA of the complementary codon that exists in this bigger subunit. Hence, two codons are held by two molecules of tRNA placed near to each other and a peptide bond is produced among them. When this process repeats, a longer chain of amino acid is synthesized. At the stop codon, ribosome releases the amino acid chain. Recap session about translation process. Translation refers to the process of polymerization of amino acid to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA, and the amino acids are joined by a bond which is known as peptide bond. Formation of peptide bond requires energy, and thus amino acids are activated in the presence of ATP and linked to the cognate tRNA by the process of charging tRNA or aminoacylation of tRNA. If charged tRNA are brought close enough, a peptide bond forms which is enhanced by the presence of catalysts such as ribosomes. Ribosome is in inactive state, exists as two subunits, that is large subunit and a small subunit. There are two sites in the large subunit for subsequent amino acids to bind and thus become close enough to each other for the formation of a peptide bond. Translation proceeds in three different stages or phases, and every stage is associated with different proteins. ATPs are the main source of energy used in this process. The three different stages of translation are initiation, elongation, termination. So this will be like after activation of amino acid. So these are the three steps to start the translation process. Let us discuss first about initiation. For initiation, the ribosome binds to the mRNA at the start codon AUG, that is recognized only by the initiator tRNA. What was the start codon? It represents three nucleotide base pairs, adenine, uracil and guanine. So this represents the start codon. Next we are going to discuss about the second step process which is involved in translation, that is elongation. The ribosome proceeds to the elongation phase of protein synthesis. During elongation stage, complexes composed of amino acid linked to the tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pair with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA, and automatically the process which is synthesized. Termination. So this will be the last final step which is involved in the process translation. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. So termination is like end. Students, in this session we study about translation process. So here it is followed by three steps. What are they? Initiation, elongation and termination. Thank you.